This is part 2 of 4, looking at chapter 15. In the first part we looked at the cell cycle. In this part we're going to focus on mitosis. Alright, so we're going to look at the mitotic cell division part of this chapter. So mitotic cell division, this is when a cell is going to divide and it's going to produce two new cells called daughter cells. And these two daughter cells are going to be genetically identical to each other, and they're also genetically identical to the original cell, which is referred to as the mother cell. So basically it's kind of like cloning. We're going to take one cell, and we're going to take that one cell, divide it in half to get two identical cells from it. So this process includes mitosis, which is the division of the chromosomes. And then it also is going to include cytokinesis, which is that physical separation of the two cells. Mitosis is used for asexual reproduction. So you can take one unicellular organism, it will split in half, and you'll end up with two unicellular organisms that are identical to each other. This is also used for growth and repair. So as we grow from one cell, we again all start off as one cell. As we grow from one cell to our 10 trillion cells, or like I mentioned, it can also be used for repair. So if you cut yourself, the cells surrounding that area will go through mitosis to repair that cut. So right before cell division, we went through that cell cycle, but remember that the cell's going to go through the G1 where it grows, collects material. It'll go through the S where your DNA is replicated and you end up with chromosomes that look like the little X's that we see up here on the slide. And those chromosomes are composed of two sister chromatids that are tightly associated with each other at that centromere. The centromere, it's going to serve as an attachment site for protein fibers called kinetochores, and those kinetochores are used to sort the chromosomes and to pull the chromosomes apart during the cellular division. So in order to pull these chromosomes apart, we're going to have a structure in the cell called the mitotic spindle. And the spindle, this is a sorting process that's going to ensure that each of your daughter cells are going to obtain the correct number and types of chromosomes. So remember, we want to make two daughter cells that are identical to each other. So each of these daughter cells have to have one of each of our 46 chromosomes that we have in our cells. So the mitotic spindle is responsible for organizing all of the chromosomes and sorting them properly during the mitosis event. So in the mitotic spindle, we're going to look at an animal mitotic spindle. We have the centrosomes. These are going to be found at the two poles, so they're going to be across from each other within the cell. The centrosomes, this is where the spindle is going to be organized. So it's going to organize all of these microtubules, which are basically really long proteins. So you have to have somewhere to organize them. So a single centrosome, most cells have one centrosome. It's duplicated at the beginning of the M phase, so during interphase. At the centrosomes, animals have structures called centrioles, which are not found in other eukaryotic organisms, but they just help to organize these different microtubule protein fibers. If you look at the image on the right-hand side, we have two poles, one at the top and the bottom. Those two poles, they have the centrosome, and because it's an animal cell, at the centrosome you'll have a centriole pair structure. So the centrosome with the centrioles are going to organize different types of microtubules. So the green microtubules, called the astral microtubules, those attach to the plasma membrane, and it just helps to keep the centrosome in place at the pole. The white microtubules are called the polar microtubules. Those ones are not attached to anything, but they radiate out from the poles towards the middle of the cell. And then the third type of microtubules are the kinetochores, which are in the yellowish gold in the image. The kinetochore microtubules are going to attach to the kinetochore at the centromere of your chromosomes. So kinetochore microtubules, those are the microtubules that are going to pull the two sister chromatids apart from each other and then pull those sister chromatids to their respective poles. So it's really important to organize your chromosomes before we actually pull them apart and create our two daughter cells. So this is basically kind of what I just explained, but now it's written down for you. So at the mitotic spindle, at those two poles, the spindle is formed from those microtubules, which are tubulin proteins, so basically long proteins. And we talked about the three types of microtubules. The astral ones position the spindle in the cell, they set up the poles, like I mentioned. The polar microtubules, they separate the two poles. Those are the white ones that radiate out. And then the kinetochore ones, the yellowish gold ones, those are the really, really important ones. They attach to your chromosomes, and then they're going to be the ones that pull the chromosomes apart from each other. Okay, so we talked a lot about chromosomes. We talked a lot about this mitotic spindle that organizes the chromosomes and makes sure that the chromosomes are pulled apart properly. So now we're really going to get into mitosis and what happens. So we talked about interphase a little bit when we looked at the cell cycle. So the interphase includes the G1, the S, and the G2. Most of the time in our cell cycle is spent in interphase. So interphase, it's a phase of the cell cycle during which the chromosomes are decondensed, so they condense down, they're replicated, and they're found in the nucleus. And then once you get past the G2, then you go into the M phase, which is mitosis in this lecture. And then mitosis, we're going to split into prophase, prometaphase, metaphase, anaphase, and telophase. And then finally, we'll have our cytokinesis. Okay, so we're going to start with a cell that has all of its DNA replicated, and the first thing is prophase. So we're going to be looking at a cell that has 6 chromosomes in it, or 3 pairs of chromosomes. So prophase, our chromosomes have already been replicated, so we have 12 sister chromatids, and those 12 sister chromatids are attached to each other, so we have 6 chromosomes. The other thing that happens during prophase is that the nuclear membrane is going to dissociate into vesicles. So basically the nuclear membrane is going to break apart to allow the chromosomes to get out into the cytosol. The chromosomes are going to become very, very condensed into highly compact structures, and this is where we can see the chromosomes using a light microscope. And in lab, we'll be looking at different cells that are in different phases of mitosis, so you can actually see the chromosomes because they are so tightly compact. After prophase, we get into prometaphase. So this is where your nuclear envelope is completely broken apart and your chromosomes are out in the cytosol. In addition, the mitotic spindle is going to be fully formed at this phase. So your centrosomes are moving apart towards your 2 poles, and then you have your 3 different types of microtubules that are being formed. The spindle fibers start to interact with your sister chromatids, and your 2 kinetochores on each of your pairs of sister chromatids are going to attach to the kinetochore microtubule on your opposite pole. So basically, every single one of your chromosomes is going to be attached to the 2 poles using these kinetochore microtubule proteins. And then here is looking at these phases and what they actually look like. So up at the top we have a cell that's actually going through these different phases, and we're looking at it with a microscope. On the bottom we have kind of the cartoon drawing to represent these different phases. So in the first column on the left-hand side we have interphase, and this is probably like a G2, really late G2 part of interphase, because our chromosomes have already been replicated during that S phase of interphase. So our chromosomes, we can see that they're still squiggly. That represents that they're not fully condensed down yet. And the chromosomes have been replicated, so every chromosome has the 2 sister chromatids. In addition to the interphase, our centrosomes have replicated, so we now have 2 centrosomes. Prophase, our nuclear envelope starts to disassociate, or it starts to break apart. The chromosomes start to condense down even more, so we're starting to actually be able to see the chromosomes. And our centrosomes are starting to move towards the 2 different poles. Prometaphase, our centrosomes are at the 2 poles. Our mitotic spindle is established, including those kinetochore microtubules are attached to your chromosomes from each of the 2 poles. So every one of our 6 chromosomes, the 3 blue and the 3 red, are attached to our 2 poles. Using those kinetochore microtubules. So that's our prometaphase. The next thing that happens, once all of our chromosomes are attached to the 2 poles, those pairs of sister chromatids, or the chromosomes, they're going to be pulled back and forth by the 2 poles until they're aligned along a plane that's halfway between the 2 poles. And this is an invisible plane. It's called the metaphase plate, or sometimes it's referred to as the metaphase equator, because it's that invisible line that's in the middle of the cell. So all your chromosomes are going to line up into the single organized row along this metaphase plate. When this alignment is complete, the cell is said to be in metaphase. And this is very, very easy to see in your cells, because all the chromosomes will be in a straight line in the middle of the cell. Once you have metaphase, then you're going to have the anaphase part. So far we basically have been setting up, we let our chromosomes out of the nucleus, we set up the mitotic spindle, we lined all the chromosomes up in the middle, and they're attached to the 2 poles. Anaphase, this is where we get to the action part. So this is where connections between your pairs of sister chromatids is broken. So this is where your chromosomes are being pulled apart from each other. Once those 2 sister chromatids are no longer attached, they are now considered individual chromosomes. So now we have double the amount of chromosomes because they're no longer attached to each other. Those kinetochore microtubules then shorten at the poles, and they pull those chromosomes towards the pole to which they are attached. In addition, the 2 poles are going to move further away from each other because the polar microtubules will lengthen and push against each other. So chromosomes are being pulled towards the poles, and the cell is pushing the 2 poles away from each other. And then once the chromosomes have reached their respective poles, they start to decondense down from their really condensed chromosome structure, and a new nuclear membrane is going to reform around the 2 separate nuclei. So here's what those 3 phases look like. So on the left-hand side we have metaphase. So you can see that all 6 of our chromosomes, they get pulled back and forth between the 2 poles until they line up at that metaphase plate, or the metaphase equator. So now the cell's in metaphase. And you can see in the image on the top, it's really easy to tell if a cell's in metaphase because they're all lined up in the middle. Anaphase, your 2 sister chromatids are pulled apart from each other, and they're pulled towards their respective poles. So now you can see each one of those sister chromatids is now considered to be its own chromosome. The telophase part, now we have 6 chromosomes on the left, 6 chromosomes on the right. They're individual chromosomes now, so they're just those straight chromosomes. The nuclear envelope starts to reform around the 2 groups. And then in addition, during telophase, you can also have cytokinesis. So telophase and cytokinesis are two separate things, but they can actually happen at the same time. Because remember, cytokinesis is the physical separation of the cytoplasm in the cell. Telophase is where the nuclei start to reform around your chromosomes. So the cytokinesis, in most cases, mitosis is quickly followed by cytokinesis. Cytokinesis just means that the two cells are physically separating from each other, and it happens differently between animal and plant cells, and that's because of the cell wall in plant cells. Animal cells, which is the one that was represented in the diagrams, we have something called a cleavage furrow. It constricts the middle of the cell, kind of like you're putting a rubber band around the middle and making it tighter and tighter until those two cells separate from each other. Plant cells, because they have a cell wall, what they do is they build up a cell plate between the two nuclei, and that cell plate grows and grows until it becomes a cell wall, and it separates the two cells from each other. So here's what that looks like. Up at the top we have that cleavage furrow forming in an animal cell. So basically you have a rubber band around the cells and it squeezes until the cells separate. The plant cell on the bottom, you have the formation of a cell plate, and eventually that cell plate forms all the way across and it physically separates your two new daughter cells. So here is a really good diagram of mitosis, so it'd probably be a good idea to kind of go over it and really understand what's happening. Mitosis, we're taking one cell and we condense down all the chromosomes, line the chromosomes up, attach them to the two poles, pull the chromosomes apart, and then reform the nuclei and separate the two cells from each other. So remember, mitosis is used for asexual reproduction and also growth and repair for cells.